Yep. Okay. Hello, my name is Christopher Mason. I'm the World Corn Professor of Genomics, Physiology, and Biophysics at Weill Cornell Medicine. And today I'm going to walk you through some of our work looking at microbial evolution in metagenomics, subways, and space stations, and how we use tools to better understand the diversity and kinds of evolution that we see, and ways to characterize it, ways to sequence it, and everything from wastewater to subways, what we see in cities, out into spacecrafts and space stations, and then looking ahead as well. So first, I'm going to start with what we really like to do is a lot of shotgun sequencing. This is mostly because we get a lot of information from the shotgun sequence data, much more so than we do from, say, just targeted sequencing. We can see plasmids, phages, human ancestry, a wide array of kinds of life and their adaptations and their annotations and what they're up to by doing shotgun sequencing. And so this has led us to a wide array of types of places where we do look at microbes, and this includes, for example, some places like we look at in the Extreme Microbiome Project, which you can see here. For example, Lake Hillier, this pink lake is a very candy pink lake, but it's one of several places we've been examining microbes and how they adapt and how they evolve in a range of places that have higher, lower, or different salinity, temperature, pressure, gravity in the case of space stations, or different pH and ionic content. And so really using this as a roadmap for how do microbes evolve, change, and really figure out a way to survive at all, and using that as a really a guide of what life can do and someday what it might do as well. So we've looked at places like Lake Hillier. This is the Candy Pink Lake in southern Australia. You can see here if you go to the lake on most days when it hasn't rained too much, there's this kind of nice pink hue. If you step into the water, as Ken McGrath did here in this picture, it actually lyses your cells because it's so painful, because it's lysing. It's a hypertonic solution on the eukaryotic cells, so it's breaking and lysing them. And if you go to the shore, you can grab these large chunks of salt that are around the shore, and you'll see creatures like Daniela salina within the water, and a range of other species. So we did shotgun sequencing as well as targeted sequencing for this lake, and published some of this work a couple of years ago, 2.5 years ago, looking at the differences of what's in the lake, in the sediment, in the shore, and then also in the water. So in particular, this is again where it is in Australia and southwestern Australia. You can see here kind of this little middle island section where you get this pink lake. And we look at the bank sediment, the surface water, and also mid-depth water, and then characterize, froze them, put them in DMSO and an ethanol, and then did targeted and shotgun sequencing as well as metagenome assembled genomes. So after profiling, characterizing these samples, we could actually see here that we picked up a wide range of archaea and bacteria, not as many viruses and eukaryotes, but that was as expected. I'll come back to some of that a little bit later. But there were differences between what we'd pick up depending on where we looked. You can see here the sediment and water were somewhat different between what we'd pick up in different areas based on shotgun sequence. But we did pick up more organisms with shotgun as expected because we have the capacity to see them with that method. But then we wanted to look at the differences between what we picked up. So first we looked at WGS, number of species picked up compared to amplicon. And we could pick up some that were consistent, like some halobacters and some halophiles. Generally, we could pick up at the top. You can see the abundance both by amplicon and shotgun sequencing over on the right we could pick up. And then we also looked at some eukaryotes where we could pick up, as I just alluded to, Daniela salina, we could pick up here with both methods and pick it up both in the water and sediment in the banks. Some organisms were particularly abundant and ubiquitous, and Salinobacter ruber is another one that likely contributes to the pink color that we could pick up with all these methods. But it does show there's, these overlaps are pretty small. So it's not as if we're picking up everything the same with each of these methods. We are seeing pretty different views of the biology that's present when we actually look at different methods. And so this is one of the points of the paper. When they agree, it's pretty consistent and explains likely a lot of the biology, but there's a lot left to be discovered in the different modalities of how we've characterized these wastewater in these samples. We did then also look, though, at what do we see for the number of biosynthetic gene clusters, or BGCs, that were picked up related to different species. And you can see all the pathways in the lower left, which is a very dense plot, but we could pick up some with both methods. And in particular, the Salinobacter ruber was one method that we could really pick up consistently, and it's likely driving a lot of the pink coloring, we think, as well, and other halophiles. And also by BGC class, we could see, for example, non-ribosomal peptides and other sequences and things, terpene metabolism, things that seem to be able to process a lot of that would be this harsh environment for these microbes. But we can and do pick them up, you can see with this method here in this paper. And so that was, one of the first, vignettes of what we looked at for some metagenomics and targeted sequencing in kind of an unusual environment. But we have since then been looking at many other environments and trying to understand what's present. So, for example, this is work from Scott Tye when he was in Antarctica here, collecting, profiling, and then sequencing some samples there. We've been trying to think of other locations where we can learn about biology. So this includes even the Gowanus Canal in Brooklyn, New York, which is a EPA Superfund site, so it's full of toxins and waste and some sewage. So it's a pretty rough place, but has interesting microbial ecology, we think. And we have gone there and done sampling and swabbing and some sequencing. This is Chandrima, as a grad student in the lab who's been out there collecting samples and sequencing and characterizing them as well with Elizabeth Hanoff at NYU. And so it's kind of a fascinating little experience. And there's other places too where you can actually do sequencing, of course, as far away as the space station. And I'll come back to this later, but this is a really pioneering experiment by Kate Rubins, who's from NASA, who did the first work of doing the dawn of genomics in space by getting sequencing to work up on the ISS. And this is the first time we got genome sequencing and assembly on the space station. And with that also included some of the base modifications because it was a nanopore sequence, where we could also see, the single molecule sequencing also showed some of these base modifications present as well, which you can see here, as well as in this paper. And so there's many places we've been looking, but for one of the vignettes I want to talk about in depth today is also in the New York City subway, which if you ride the subway, you will see people sometimes ride with a cloth like this, where they're terrified it seems to touch the steel railing. You'll also sometimes people upgrade and wear, a glove, latex glove, which makes much more sense because the aperture of holes in this paper towel is probably much larger than the microbes, so it probably doesn't even really have that much of an effect. Whereas the latex glove probably does a pretty good job. But you can see people ride the subway and they're always, afraid of what's there or curious what's there. And, even during the pandemic, especially people were worried. But my interest in this sparked a lot from early work in microbial sequencing. But also at one point, our daughter, who at one point you can see her very cute with a ice cream cone, did grab a pole and even lick a subway pole at one point and put her hands in her mouth. And so, if any of you are parents in the audience, there's this moment of complete parental terror where you realize there has been a microbial exchange, and you really wanted to understand what it was, what had happened, what had transferred between the pole and the mouth, and the mouth and the pole, and what's present in this ecosystem that's shared by millions of people every day in mass transit systems in general. So this sparked a big project in the lab to go out and just swab and sequence and find out. So this is something we call the microbiome of the metropolis, or the metropolome you can see here, as it was coined by the New Yorker. And wanted to go out and actually really characterize what was present in the city. So we went out across all 5 boroughs of New York City, swabbed, annotated, did a timestamp. You can see your data entry as well. Uploaded that sample and did about 1,500 samples of shotgun sequencing, generated about 10 billion reads, trimmed them, compared with Metaflan and BLAST, and looked at the species that were detected with both methods. And then we combined those two results, which you can see that we found thousands of species, but of all the billions of sequences, on average, half of them mapped to no known organism. There was no annotation. They didn't map to a known reference. They were really kind of this new map of what was present in these microbial ecosystems. Many of the reads did map to bacteria and viruses and some eukaryotes you can see here. So there were a lot of expected results, but it really showed that there's this vast unknown world under our fingertips. Where again we can see density of some species we do know, like Pseudomonas, for example, and that genus. But a lot of the reads map to these unknown areas, which is actually is very common for metagenomics. It's because our databases are so incomplete. This just shows how much is left for us to discover and characterize in these samples. So we did go out and look at the Gowanus Canal. You can see here's another shot from the canal. We've also could see there it has a lot of methanogens. You can see there's part of this is probably because of the sewage that's coming out from that system. We can pick this up here. But again, this big question that we've been pleasantly haunted with since 2015 is who is there and what are they making? In particular, what are these organisms doing? Could we predict their biochemical capacity? Could we characterize them better? Can we better understand their biology, their ecology, and how they interact with each other? And so what we could see is if you look by species types, you can see there's varying densities across the city. But one that really stood out in this first paper that we looked at in 2015 was Pseudoaltemonas, for example, only showing up in this station. And so we thought, well, that's really interesting, but why is it showing up only there? It turned out this was the station that was flooded by Hurricane Sandy. It had these 10 specific species we didn't see anywhere else in the transit system. And this was a system that actually, if you remember Hurricane Sandy, this was now, almost, back in 2012, knocked off government websites. It was a huge hurricane that actually came up into New York City. At some point, it felt bad as if all whole streets were going to be submerged. it was not quite this bad. This is actually from the day after tomorrow. This is not a real picture, but this is a real picture. This is basically the transit system after the flooding. We could actually see here, differences where we could have these grayed out stations that were not accessible, that were either inactive or flooded or had severe damage and had to be repaired and cleaned out. And so this was a, pretty significant storm that hit in 2012. But we're able to work with the MTA, the Transit Authority, to get samples of the station before it was actually opened back up to the public. And we wondered if there was a persistent molecular echo of the cold ocean water, in this system. And so we were able to actually go in and swab, and we could see interesting species like Schoenella frigidmarina, which was previously thought to be an Antarctic species. And here, actually, we know it can make eicosapentaenoic acid, which is something that you find in your diet normally from eating fish. It even has, a beneficial feature in the diet. So we could see this is, in some sense, a good organism possibly there that might even be helpful because, there's some evidence that even reduces risk of suicide likelihood. So not all is bad that's down there. And so there's really interesting sort of echoes of what's left behind. And so this was in the subway, but we're going to think, well, where else can we look, on Earth or anywhere beyond? And in particular on Earth, can we think about what's happening in sewage? And here we've done a lot of work with Helena Stella Gabriel and George Grills and many other teams at the University of Miami and part of the Metasub consortium to sequence and characterize the wastewater across these different systems. So, we could see, and I think it's been, published by several groups that you can see this correlation between sewage and population metrics. And we can see these clear differences over time that what's present for the positive caseload in the population can mirror and recapitulate be recapitulated what's in the wastewater. So it's really interesting map to see what happens here in the system. And we published this last year as really a geospatially resolved public health surveillance example from this data set. So in particular, we've been sequencing across 2021, 2022, and into 23, even up to this day, some other sites. But we could actually see the emergence of different alpha, beta, epsilon, omicron strands. We could see the allele frequency of these clade defining variants show up over the years, which we could pick up again and again. And you can see them here as they're pulsing. We could also pick them up, not just viruses, but eukaryotes, bacteria, archaea. We could see, a wide range of densities of organisms and a lot of different kinds. But, the shotgun RNA sequencing gives us this real nice pan-domain surveillance of all the microbes that are present. And so you can see these different wastewater treatment areas. And some of them, for example, we could pick up fungi like Fusobacterium. We could pick up Saccharomyces. A lot of them are rare, but we can pick up some pretty common species for fungi. And for bacteria, we could pick up like things that are even in plants, like Xanthomonas. We could pick up, but we could pick up a lot of Previtola. We could see even in some cases some sulfur-related bacteria or very human-associated bacteria, or even sewage-associated bacteria, and in some cases even some salmonella traces in certain sites. So really interesting view of the biology of these sites. And what's kind of fun is we could even look for new pathogens that were uncommon, but for example, looking for HEI virus. This is an email with Mark Johnson saying, oh, do we see it in the data set? And indeed, we could pick this up, HEI virus. We could pick it up here up in the upper right. So we can start to look for others, viruses that are circulating and trying to figure them out, which we're now doing with the Human Virome Program at the NIH, is trying to figure out what happens when these normal viruses are infecting otherwise healthy people and how they move around the country and around the planet. Now, what we showed in this paper from Dr. Tierney was that we could find, of course, known viruses like this Aichi virus, but also could pick up, this norovirus that's here. We could see that the query genome was aligning quite well within the clades of the known phylogenies, but also we could pick up other species that are health-related, like Campylobacter, Heliobacter, Clostridium. So we could start to pick up some of these really intriguing species that may be related to other facets of human health and pick them up differences. You see in the lower left what's the difference between the hospital, the campus, the dorm. We do see density and differences depending on where you were on campus for the University of Miami. Also kind of interesting to see these regional differences. And so one particular example of this actually is we could see, again, the campus bays in the dorm, the hospital. You can see the hospital back here. The hospital has this little burst here of Klebsiella, which you don't really see anywhere else. You can see this down here in the lower left. And so it shows that there's these, regional differences. We can pick up differences of where you are in the city, are you in a hospital or in a dorm? There are these differences that show up, as expected in the sense these are more pathogen-related hospital and infection-related that you do see at the hospital. You can see here in the lower left. And we also could see, for example, well, does this link to the antibiotic resistance or AMR genes in general? And you can see here, we can see it's significantly higher in the sewage in the hospital than we see for other locations. And it's correlated generally with prescriptions. It's a noisy correlation, but it's a positive correlation. In general, like we see what's being prescribed and what are we picking up for the resistance markers in the wastewater. We do pick that up as well. And so this really gives us kind of like a metagenomics of sewage or MetaSew, which links to a larger project, which is born out of some of the subway work called MetaSub, which is looking at the genomics and subways, design of subways in urban biomes, with three global goals, which is to look at these metagenomic maps, which could be useful for discovery, but also forensics, looking at AMR markers and how they change, and also new biosynthetic gene clusters, tracing them as well. So what's interesting is if you think about where we've used this before, historically, even rapamycin, for example, was found by doing an expedition to Easter Island in 1964, just digging in the soil. So this big question of natural product discovery, we often think of it for soils, but we could look at it really anywhere, and we can obviously predict them from like Hillier or other places. And so this is one of the big goals of the entire consortium is to say, okay, well, if you found all these active compounds from microbes, a lot of them are made by actinomycetes, some are fungi, some bacteria. But there's a lot left to discover in the world all around us, in some cases, again, literally under our fingertips. And so for Metasub, one of the first big data sets released a few years ago was looking at what's present across the world. In this case, we did about 6 million reads per sample and wanted to see what was there. So in particular, we did sequencing, alignment, and also de novo assembly to characterize what was present in these samples across the world. So what we found was basically a threefold increase in the size of the tree of life, including 11,000 new viruses, again, published a few years ago. And if we do a little bit of a deep dive in the paper, we could see that getting up to several thousand samples, we were seeing, beginning to get to the plateau, but still more discovery left for sure if we were to sequence more samples. And across these almost 5,000 samples, we could see that a lot of them were generally rare. You can see here, but there were some species that were present consistently across almost every single sequence sample in the prevalence. So there were actually, 61 species present in 95% of all the samples. There were then, some ability to predict where you're from. So each city, even though they're processed the same way and sequenced the same at the same time, some regional differences as well. We could predict maybe where you're from. And if we look at species that were present in 97% of samples, there are these 31 species that we showed up again and again and again. And you can see some of them are commensal, some of them are from biofilm, some don't, some are gram-positive or gram-negative. But a lot of them are ones that make sense, like Staph epi, Staph hominis. These are human skin associated microbes that are, very expected. Some Cutibacter acne, of course, you'd see also on human skin. And so some of these made sense, but other ones, like a lot of the Pseudomonas petita, Staphytropomonas, might be other species that have seemed to have adapted to this urban environment, or at least are, tolerant of it. And we can pick this up, for sure, consistently, again, again here in the environment. And so we could also see the AMR density was quite different between class and also what city you came from. So some of the cities on the right had orders of magnitude more versus less AMR markers, as antimicrobial resistance markers. And here we wanted to see, could we recapitulate that metric of what is the population taking for over-the-counter antibiotic use? And we can actually see, again, a positive correlation, even if it's noisy, of the dose per thousand individuals and the density of these AMR markers, both on chromosome or plasmid annotation. It's work from Patrick Lee in the consortium. And so indicate again, we can use the wastewater data to somewhat reconstruct what the population is taking and doing in terms of antibiotic usage. So again, before you get maybe alarmed of like what's there, this is actually much less than what you'd see in a cow gut sample or soil in terms of the density of the AMR marker. So it's not as if the subways are terrifying places in any regards. It's much less of the AMR density than what we've seen elsewhere. And so we did kind of characterize these AMR markers, but also found over 800,000 new CRISPR arrays in this data set. So many of them were these spacer hits per megabase, much more pronounced in the metasub viruses. We could see distribution of these viral contigs and density across the world. So picking up a lot more of the CRISPR activity as well. It's basically, we're getting an echo of what is it that the microbes themselves are battling when they're in the environment. And another great piece of work by Gunnar Reich and some of the teams there, in ETH Zurich, and this is Andre Callas as well, made this map for part of the paper, which we can type in any sequence and see where does it appear around the world, this global KMER index, which you can go to this website here and play with the data set more. But it gives you a way to track, basically what species are co-occurring with each other, which molecules do we see again and again, even if we don't know necessarily yet what something is doing, we can see what is it always coming with in terms of its coincidence of these other molecules and other sites around the world, this sort of global KMER index. And so what's interesting is we've done this, obviously at Cornell, but also there's an outlay of GeoSeq Foundation where you also now overlay climate data, health variables, and can start to begin to, build models of what's present. So for example, this is something we call the Watchtower Network, and this is called GeoSeek, where you can see historical data, mosquito incidents, and now working to overlay this kind of information to build data models, for example, of dengue in Brazil, and working on this now with the GeoSeek Foundation and with Biosha as well, with a conflict of interest, one of our own spin-out companies, but doing a lot of work here to build these maps and visualize what's present across these databases. And what this gives us now is actually an ability to have planetary genomics. We can actually see what's present quite literally around the world. And we can even start to get at interplanetary genomics, or getting at least at a low Earth orbit. And the first chance we got with this was the Inspiration 4 crew, where we actually swabbed 10 different regions of their body to see what changed during a 3-day orbital spaceflight. And what's extraordinary about this is we could DNA and RNA sequencing and start to see, well, how much do they get together? How much do the microbes converge or not in just a 3-day mission? And we hypothesized we'd see something similar to what's been seen before for skin microbiomes after sports, that they get more similar with each other in that tight environment. And so we again did metagenomics, metatranscriptomics across the whole crew as well as the capsule, and looked at the differences over time of these different samples to look at these time points and samples. And we could actually see that the armpit was one of the greatest sources of biodiversity, and there was some in-flight convergence where the differences or the beta diversity did decrease during the mission, which we can see here. We could even see some evidence of pizza and maybe even November cold that showed up, a rhinovirus at one point we could pick up in some of these samples. You can see here. But we can see what changed actually. A lot of them were Caulobacter. We could see those are the ones that changed a lot in flight, and there's a lot of adjustments and dynamics happening when we see for these species. And also we could see in some cases there's a transient decrease or increase or long-term increase depending on what species you looked at, and for mid-flight versus pre and post-flight for these microbiome dynamics. And so really interesting dynamics we can pick up again and again. Again, some of them are ones that are human associated, or ones that are E. coli, for example, or Corynebacter, but other ones seem to be much more transient that we see picked up even coming from the capsule itself. So we published a lot of this work last year. This is a large set of several dozen papers from many teams working together in the Space Omics Atlas, and you can now go and plot out some of these data sets and also gene expression patterns as well from some of the attack-seq and gene expression data. And there's also a browser where you can go in and look at individual species across the crew members and see, what's different between the DNA and RNA sequencing and different regions of the bodies for these different crew members. So it clearly shows that there's some spaceflight effect on the microbiome and in transmission. So another example we've looked at this in space stations was looking at Acinetobacter pittii and how it's changing in the ISS. And this is work with Katsuri Venkateswaran or Venkat, where we looked at longitudinal profiling of microbes. And here we actually did this as a case study to see, well, what do we look at? What can we see for the phylogeny? What's the succession across time on the ISS? And how much do we see some of the, when we see AMR, when we see resistance, what's driving it potentially? And so what's interesting is we looked at, all these different strains. You can see here in blue, these are the different sort of microbial tracking strains, and all the other ones above and below this on this heat map show you basically all these different Earth-based strains. And we could see basically there's this presence in sort of an ISS core genome that was enriched over here for these stress response genes, which we didn't pick up in any of the other, rarely picked up in some of the other terrestrial strains, and shows there's this interesting enrichment for this kind of stress response, that even though they were not antibiotic resistance genes, they're more stress response genes, they do seem to confer maybe some resistance to the antibiotics for these strains. So interesting to see, this sort of different outgrowth of a different clade that's has after, these species have been in space in some cases for years, and they have started to diverge from the Earth-based and terrestrial strains, which is kind of fascinating to see. So we did this, here looking on the space station, but we've also worked with JPL to look what's in the clean room and see, as you build spacecraft, you have to think about, well, what's on that system, and you think, okay, is it someone's getting excited, they take a selfie because they're building a robot that's going to get sent to space. But in this case, the rover, when it went to Mars in 2021, it landed. There's a Mars sample return mission where eventually we're going to get samples back to Earth, and this is what it looks like on Mars. At some point, this has been delayed a little bit recently, but at some point we'll get this Mars sample return. And then you have to wonder, well, do we get microbes from Mars? Is there life on Mars, or do we have to control for anything we saw in the clean room, which is indeed what we've been doing. So we built out this metagenomics framework for planetary protection to characterize what's present and what could possibly survive, or even be, contaminants that you could correct for and how they might survive. So this is one of the sort of projects that's ongoing for building this kind of planetary scale frameworks for metagenomics, and then deploying them for examples of cases where they could be useful for filters, much like this one. And so again, this is K-mer index that as soon as we see anything that we think might be life on Mars, we can quickly characterize it, annotate it, and search against this database to find any possible microbe that may have been seen on Earth. And if there's nothing that's been seen before, then I think you could start to say, okay, we think we see a microbe, or at least a K-mer or some fragment of DNA that has never been seen before on Earth. And again, we see that a lot even in the subways, but we could see is it radically divergent? Does it look like nothing at all and keep changing the filters? But at least gives us this first pass ability to look at what's present in the sample and compare it even between planets. As you can see, here potentially you can see these differences. And so, begs the question long term, we've been thinking a lot about and working on ways to think, could we make plants and animals that could survive on Mars? And could we find organisms that can, survive, for example, in high levels of carbon dioxide? Indeed, we can find, for example, cyanobacteria that can absorb carbon quite quickly. And this is work from Braden Tierney in the lab and also collaboration with Max Schubert and George Church's lab. So, we can find other places where organisms have these unique abilities and capacities, and we've also been using those as lessons to try and find organisms that could grow in lunar regolith and start to plan these out as well for some work we're doing here with NASA. Again, the long-term goal is to make, the moon is someday make Mars be self-reliant. So you could have, food and production strategies. You could have backup capacity. You could make, essentially reclaim liquid waste. You could have photo autotrophs. You could recycle all of your solid waste. And at some point have go from tens to hundreds to maybe thousands of people living on the moon and Mars. And this will take really a lot of our work in mapping, annotation, characterization of microbes and getting them to be these self-reliant systems. But it will we can do some of it on the space station today, for example, but it's going to take a lot of optimization, curation, and perfection to get us to that next level where we could have basically planetary liberty where you can go to either Earth or Mars or the moon someday. And we're also even looking at, for example, interesting missions now enabled by the Starship or these high velocity rockets where you can imagine going past Mars and then Venus and then back again all within about 18 months. And so you can start to, we've started to plan out actually some of these really interesting and ambitious missions to go across multiple planets and leverage some of these new tools. And along the way, if you're passing by Venus, we can also start to look at what might be causing, some of the phosphine to appear in the clouds and why is it that we're seeing possible activity? Could microbes even survive in sulfuric acid clouds? Probably not, but maybe. So it's really great work with the Seeger lab and also with Hannah Burgess in our lab. And so with that as a backdrop, I want to think about this for the very, very long term and use these genetic tools to help us, understand and live and survive on this planet and someday, hopefully, eventually others. And here's a shameless little plug for a book I talk about this more in depth. And at the end, I want to thank everyone in the lab who makes this possible, who's extraordinary. I'm honored I get to work with all these extraordinary people and scientists, engineers, and passionate artists in some cases. And then thanks to funding and support from especially from World Quant, the NIH, NASA, coordination for a lot of these missions with SpaceX and NASA and other flight providers. And thanks to many colleagues, friends, collaborators, too many to be named here, but here's a brief list of many of the great people and friends who've made this possible. Again, funding and then finally the patients, the crews, their families, and all the data, sample, and resources and that they leave for us to study and to learn from. Thank you very much.